This video will demonstrate a prototype implementation of a personal genetic locker. The technologies used for this prototype are the FairData Point and the MyConsent system. The process starts from a personal locker maintainer perspective. In order to use a personal locker, it first needs to be populated with data. Let's create a catalog to organize different datasets of personal data. The metadata description of the catalog requires a number of descriptive fields. The title is used for human recognition. The version number is an arbitrary identifier to keep track of versions. The publisher is responsible for the published data and is described by a URI and a label. The theme taxonomy is a URI which points to the set of themes for the dataset the catalog contains. From the Share tab, the metadata can now be published in FairData Point. The catalog metadata is published under a unique identifier. When the catalog metadata is published, a link is made between the FairData Point and the MyConsent system. Let's create a dataset metadata description next. The metadata will provide specific information about the dataset. The title, version, and publisher fields are defined as before. The themes will provide computer-readable descriptions of the dataset. The link to the parent catalog is added to enable bidirectional traversal through the metadata. The dataset metadata can now be published in the FairData Point. The metadata is published under a unique identifier. When the dataset metadata is published, a link is made between the dataset metadata in the FairData Point and the data record reference in MyConsent. This results in a token, which will be given to the owner of the data. To complete the Fair metadata, the distribution metadata needs to be published. This metadata is a description of the actual file, its format, and other properties. The title and version number are provided as before. The license under which the data is available is provided. A link to the download location of the file is provided. The media type of the file is provided. The link to the parent dataset is added to enable bidirectional traversal to the metadata. And now the metadata can be published in a FairData Point. It's published under a unique identifier. Using the token we received before, the owner can now take control of the data in MyConsent. Let's register an example user in the MyConsent system, so the user registration flow can be demonstrated. When logged into the MyConsent system, the user is presented with an overview of his data. There's no data linked yet, so first a new data source needs to be linked. Providing the token generated when the data was deposited, the user confirms the link and keeps the default permissions. At any time, the permissions can be changed. The user is again presented with an overview of his data, and then switching to the Consent tab, he sees no requests as of now. Let's switch to the researcher perspective, trying to access the data. The researcher has found the FairData Point through one of the Fair search engines, and he sees the catalog with personal genetic data available. Accessing the catalog will present the catalog metadata. The researcher sees the theme taxonomy, which is in his field of interest, genetics. He sees a dataset for personal genetics data. When the researcher is trying to access the data, the FairData Point first requires him to log in. The authentication scheme is provided by ORCID. The researcher authenticates himself using his ORCID credentials. and is then redirected to the FairData Point again. The FairData Point checks the consent status of the data owner and notifies the owner of the data about a new request. Now let's switch back to our data owner role. When logged into the My Consent system, he can now see a request in the consent view. A description of the request and the study it is associated with is presented. The owner can now make a decision to consent to his data being used or to deny the request. For this demonstration, we'll provide consent. Note that at any time the owner can change his response and deny access to the data. When we switch back to our researcher, he now will be notified of a new response to his request. When he goes back to the dataset and tries to access it again, he's once again asked to authenticate himself. And since our previous ORCID session is still active, we are automatically redirected to the data. Since consent is given, the researcher now has access to the distribution metadata. In the distribution metadata, a secure link is provided to the download location of the data. When the researcher accesses the data link, the consent status is checked every time. If the consent is still active, the researcher can now access and use the data with the consent of the patient. This concludes our demonstration of the FairData Point integration with the My Consent system.